Okay, so Assalamualaikum warahmatullahi wabarakatuh and hi. So today we will continue with the topic meiosis. So meiosis is the process of cell division that occurs in reproductive organs to produce the gamete that contains half number of chromosome haploid of the parent cell that is diploid. So here if you look at the diagram here, okay, this is the parent. So in parents we have 4 chromosomes, for example. So it will produce the daughter cell that have 2 chromosomes, which it is half of the chromosome of the parents. So here the parents we call as diploid, while the daughter that is half chromosome number we call as diploid. And meiosis only occurs in reproductive organs. For both humans and animals, meiosis occurs in the testes of male and the ovary of female. So, the need for meiosis. Why meiosis is important? Because it will undergo the process of gametogenesis to form the gamete and to ensure that the diploid chromosome number of organism is always maintained from one generation to the next. Okay, for example, human will have 46 chromosomes in total. So, if a male that have 46 chromosomes undergo meiosis to produce sperm. Sperm is daughter cell. So it will produce sperm that is haploid of the parents. When the parents is 46, so the sperm will have 23 chromosomes. It is the same with the haploid egg, which in female reproductive organ, female that have 46 chromosomes, when it produces the daughter cell that form into egg, the daughter cell will have the half number of chromosomes, which is 23, half from the 46. So the 23 chromosome of the sperm, when it fertilizes with the 23 chromosome of egg, it will form back the 46 chromosome that will form a diploid zygote. So that is how important that we need to maintain the diploid zygote, which the sperm and the egg needs to be the haploid of its diploid parents. Okay, so please find what is type of gametogenesis for male and also female. Okay, next, the need for meiosis. So meiosis produce genetic variation in the same species. So how it produces genetic variation? Later we will look at the process. So meiosis is important because if humans have only the same structure, the same skin, the same shape of face, the same hair color, the same hair type, so there will be no variation among one species. So that is how meiosis function to produce the variation. So if you can see the picture here, whom family is this? Can you recognize whom family is this? So, can you notice what are the differences or variation that occur among the family members? Can you notice what are the differences or variation that occur among the family members? So, how about the variation among this family? Okay, if you can see this family of dog, you can see they have some have curlier hair, some have a bit straight hair, and also the color of the fur is also a bit different. Okay, this one is a bit darker, while this one is more brighter. Okay, so that is how we can see variation in one species. So for meiosis, it is divided into meiosis 1 and also meiosis 2. To remember, we can remember it through P, which P is for prophase, M for metaphase, A for anaphase, while T for telophase. So if you can see, between the meiosis 1 and meiosis 2, both have the prophase, metaphase, anaphase, and also telophase. What makes it differ is that for meiosis 1, all will have 1, prophase 1, metaphase 1, and for meiosis 2, all will have 2 at the end, prophase 2, metaphase 2. Okay, so next we will look at the meiosis 1 first. Okay, if you can see, this is the stages. We have prophase 1 followed by metaphase 1, followed by anaphase 1, and also the last one is the telophase 1. The aim of the meiosis 1 is to form two daughter cell that is haploid. Okay, so meiosis 1 cannot stop there. It will be continued by the meiosis 2 to form the complete haploid, 4 haploid daughter cells. So let's look at prophase 1. The chromatin shortens, thickens, and forms visible chromosome. Okay, if you can see here, the chromatin, if you can remember in the topic nucleic acid, the chromatin is the one that made up the chromosome. So when it shortens and thickens, it makes the chromosome structure to be visible under the light microscope. And then the pairing of homologous chromosome forms bivalent or 4 chromatids for each homologous chromosome. Okay, in prophase 1 only, the homologous chromosome will be paired up. So how do we recognize the homologous chromosome? As been mentioned in mitosis before this, okay, homologous chromosome is the one that have the same structure. If you can see here, the red and also the blue one, the length of the upper arm is both the same, and the length of the leg is also the same. And if you can see the band, okay, this one is what we call the band. Okay, if the letter is different, whether it's capital or not, it doesn't matter. The thing is that the letter is the same. If the first band is A for both chromosomes, so it is the same. And if the second band is B for both, so it is the same. Okay, so that is how we detect them as the homologous chromosome. And also, if you can see inside the cell here, okay, this is the pairing of the homologous chromosome. You can see that the structure is the same. And also, it forms bivalent. Bivalent is what you call heterochromatic. If you can look at here, okay, homologous chromosome. This is the first chromatid, second, third, and also the fourth chromatid. And also in prophase 1, we have crossing over occur at the point of chiasma. Okay, the crossing over will cause the exchange of genetic materials between the non-identical chromatid. So if you can see here, okay, this is the homologous chromosome. Okay, so this is the chiasma, the point of exchange. Okay, if you can see the purple, when it crossing over, it will get the color of blue. That show the exchange of genetic materials. Why it called the non-identical chromatid? If you can remember back in mitosis, I had explained that, okay, for example, in one chromosome, the dark blue, if you can see the two legs is what we call as the sister chromatid. Okay, and also in the light blue, if you can see the two chromatid, the two leg is what we call as sister chromatid. But between the different chromosome, we call them as non-identical or non-sister chromatid, even if they are homologous chromosomes. So when they crossing over, so the point here is what we call chiasma. And you can see that this denotes the exchange of the genetic materials, causing the variation that occur in the species. And at the end of prophase 1, the nucleus membrane and nucleoli start to disappear. Okay, this one is the nuclear membrane, the grayish one is the nucleoli, and both central move towards the opposite poles. Okay, the centrals is actually both at the same place, but at the end of prophase, it moves to opposite to each other. Okay, the centrals also can be at the left and at the right, as long as they both are at opposite poles. And the spindle fiber form from centriole. So this is the spindle fiber. So next is the metaphase 1. The homologous chromosome arranged at equatorial plane. So equatorial plane also we call as metaphase plate. Okay, both is the same. So if you can see here, the GIF here, the homologous chromosome line up at the center or at the equatorial plane. And for each pair of homologous chromosomes, one chromosome attached to spindle fiber from one pole, while another chromosome will attach to spindle fiber from another pole. So if you can see here, this is the pair of homologous chromosomes. We only have two poles where the centrioles exist there. So the centriole at the first pole will attach to the first chromosome in the homologous chromosome. And the second pole will attach to the centromer of the second chromosome, meaning that if you can see here, okay, right here, in mitosis, in one chromosome, both centromere been attached to spindle fiber. But in meiosis, because of its pair up, so only one centromere of the first chromosome attached to the first centriole or spindle fiber, and the second, or at the back here, the blue one, the centromere attached to the second spindle fiber from the second pole. Okay, you can see here. So that is how the spindle fiber attached to the centromere during the metaphase, making it to arrange at the equatorial plane. So for anaphase, the spindle fiber contract or shorten. If you can see here, when it shorten, okay, it will attract it will separate the homologous chromosome towards the opposite pole. Okay, depends on which spindle fiber that is attached to its centromere. And each chromosome still exists as a pair of sister chromatid. Okay, remember sister chromatid is the two that is in the same chromosome. Okay, for example, this chromosome, two legs is the sister chromatid, and it's still attached at the centromere. The centromere is not split up because only one centromere that's been attached to spindle fiber. Okay, for the telophase, the chromosome arrive at the opposite pole. Each pole contain haploid. Okay, this one become haploid, but only two daughter cells will be formed after cytokinesis. The spindle fiber disappear, and also the nucleoli reappear and nuclear membrane being formed. So let's move on to the meiosis 2. So meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and also telophase 2. So what is the aim for meiosis 2? We want to produce 4 haploid daughter cells. For meiosis 1, we want to produce 2 haploid. But meiosis 2 is the continuity which it will form 4 haploid daughter cells at the end. So let's look at prophase 2. Okay, the nucleoli and nuclear membrane start to disappear. Each chromosome is made up of sister chromatids. We no longer call them as homologous because they had been separated. Now we call them as chromosome or sister chromatids that is joined at centromere. So spindle fibers start to form, and also the central will go to the opposite pole. For metaphase, chromosomes are arranged randomly at equatorial plane. If you can see, okay, it arranged at the equatorial plane or the metaphase plane. Each chromatid is tied to the spindle fiber at the centromere. If you can see, the two legs have its own centromere. So both centromere of the chromosome will be attached to spindle fiber, not only one centromere as in metaphase 1. Okay, because in metaphase 2, only one chromosome being aligned at one time. But for the metaphase 1, the pair of homologous chromosomes is being lined up. Okay, so metaphase 2 and when centromeres separate. Why we call them as randomly? Because this position can be switched, where the leg with the blue can be at the left and the other will be at the right. Okay, it is randomly arranged. For anaphase 2, the sister chromatid centromere starts to separate and move towards the opposite pole. Okay, if in meiosis 1, we call them as homologous chromosomes separate, but in anaphase 2, we call them as sister chromatid separate and move towards the opposite pole. And lastly, the telophase 2, the chromosome arrive at the pole of the cell. The spindle fibers start to disappear. The nuclear membrane and nucleoli reform, or it begins to appear back. The number of chromosomes for each daughter cell is half of the parents. Okay, because the parents will have 4, for example. So this one we have 2 for each daughter cell. And the telophase to end with cytokinesis. Okay, it will produce 4 daughter cell that is haploid. Haploid meaning half chromosome number from the parent. So the genetic content is also different because of the crossing over that occur during the meiosis 1, which is in prophase 1. Okay, if you can see, this one is the mixture or the exchange that occur. So the haploid cell will develop into gametes. Okay, another thing that is important is meiosis do not involve fertilization yet. It is only to produce the sperm or the egg for human and also animal. Okay, but for plant it will produce it will occur at the anther and also the ovary of the plant. So this is summary of the meiosis 1 and meiosis 2. If you can see, in prophase 1 the crossing over occur. In prophase 2 there's no crossing over occur. In prophase 1 there is a pair of homologous chromosome but not in prophase 2. So in metaphase 1 the pair of homologous chromosome will line up, but in metaphase 2 only the chromosome will line up. Okay, there's no pair. For anaphase 1, the homologous chromosome is the one that is separated and moved towards the opposite poles. But for anaphase 2, the sister chromatid been separated, not the homologous chromosome. Because of, you can see the spindle fiber attached to both the sister chromatid. That is why both sister chromatid been separated. But for anaphase 1, the homologous chromosome, one will be attached to the first spindle fiber at one pole. The other pair attached to the spindle fiber at the other pole. That's how when it shortens, it will separate the homologous chromosome. And for telophase 1, we will produce 2 haploid daughter cells, but for telophase 2, it is the complete set, which is 4 haploid daughter cells. So that is the detail for the meiosis 1 and also the meiosis 2. And again, remember that this is to produce the sperm and the ovum or the egg for human and animals. It is not yet involved the fertilization. Okay, so that is all. Thank you.